Welcome to the third CIBM Breakfast and Science seminar series. It is my pleasure to introduce you to our first speaker, who is Cristina Cudalbu, Research Staff Scientist and 9.4T MRI Operational Manager in the CIBM MRI EPFL section. She'll be giving a talk on translational non-invasive neurometabolic studies during chronic hepatic encephalopathy in developing brain, from the in vivo rat to the human brain. Thank you, Cristina.
Thank you very much for the introduction. Today is a special day. It's a special CIBM Breakfast and Science. So I will start differently my talk. Hope you will like it. So today's talk is about translational non-invasive neurometabolic studies during chronic hepatic encephalopathy in the developing brain from the in vivo rat and the human brain. So I will start by showing you why it's translational. It's translational because we are using 3D organotopic brain cell cultures. Yeah, just one question. Do you see the slides?
Yes.
Yes.
Perfect.
Yes.
Yes. So we are using these 3D organotopic brain cell cultures, which were developed in the CHUV by one of our collaborators. You will see later. So we are using them to mimic a diseased brain. So they're very similar to a brain. They have all the type of cells. They don't have the blood-brain barrier, however. We are scanning a rat model, and we are doing MRI and magnetic resonance spectroscopy in children with chronic hepatic encephalopathy. Regarding the collaboration, there are a lot of collaborations that were established during the years since we are working on this project. And the main collaborators are Olivier Bresson from the CHUV and Valérie McLean from the HUD. Without them, I wouldn't be here. I wouldn't present this. So thank you very much. Their teams, like Kasia, who's working with Olivier now, like Mark, two Marks, the technicians of Olivier, one who is leaving today, he's retiring, and the other one who's starting. Valerie with her team, Dario, Sarah, and all the other people from the HUG, like Anne-Laure, who is doing the histology of our livers, like Christiane and Graziano, who teach us how to do the surgery in the PAPs, and Sylviane, who's following all the studies that we are doing in children with chronic HE. Then we are collaborating with the lab of behavioral genetics for the behavioral studies, with Jocelyn, for instance. Then Siemens helped us to process the data using their MorphoBox, the 70 data acquired on the children. Then people from CIBM, like Li Jing, who was there all the time for the 70 scans, Ileana, Sandra, who was there all the time to help me prepare the video for the kids, and from LiveMed, Bernard and Emma. So today, the talk is divided in 3 parts. I will introduce you what's chronic hepatic encephalopathy. We will try to see the results and see why the developing brain is different compared with the adult brain in a rat model of chronic hepatic encephalopathy. And I will show you some preliminary results in the neurometabolism, so the brain metabolism in children with chronic liver disease. So chronic hepatic encephalopathy. It's described as being a spectrum of neurocognitive signs and symptoms arising in the settings of chronic liver disease. It was divided in 3 types based on the type of liver disease and based on the acuity. So we have the acute liver failure, we have the portosystemic bypass, and we have the type C, which is due to chronic liver disease. It can be severe fibrosis or cirrhosis. So this is what we will be looking into today. And all of these 3 types of hepatic encephalopathy, they have a common feature, which is the hyperammonemia, meaning that the liver is incapable to detoxify the ammonium. There is a high ammonium load in the blood, which is reaching the brain afterwards. So this is considered as being the main toxin. So patients with chronic hepatic encephalopathy, they suffer from an area of cognitive and motor deficits. They can which can lead to coma and death of these patients. The hepatic encephalopathy is ranging from minimal to overt. And the causes, as I said, it's insufficient liver functioning, which leads to many toxins in the blood, like ammonium, bilirubin, bile acids, and other more. Sorry, and we have the liver inflammation, which leads to systemic inflammation. The pathophysiological mechanisms in the brain, there are some which are known, some which are still in debate. So we know that there is an increase of the glutamine because of the detoxification of ammonium, who takes place in the astrocytes through the glutamine synthetase enzyme. Other people saw it, other labs, we saw it also. This glutamine leads to a secondary osmotic imbalance, which is translated into a decrease in the inositol, taurine, total choline. However, we don't know what brain regional metabolic changes occur, meaning we are wondering if there is a brain region which is more vulnerable than the others. It's something that is not yet known. And we don't know how the morphology and the function of brain cells is affected by these changes, by the increase of the glutamine and by the increase of ammonium. Do we have a low brain grade edema at the end or not? We are still not sure if we have an oxidative stress, even though our results made by Kasia in the lab show that there is. We don't know yet anything about the mitochondrial permeability transition. We know something, but it's not yet sure. The neuroinflammation and its contribution to the chronic HE, it's not yet clear. And again, the energy metabolism disturbances. So the molecular bases are not clear, and the animal models are very useful in understanding all this. And in our lab, we are studying all these different molecular mechanisms in type C HE. So what makes adults and children different? So adults, they become sick because so the causes are hepatitis or other viruses, the alcohol abuse, a non-alcoholic fatty liver disease, and primary biliary cirrhosis. The liver transplantation is the only real treatment. And it was shown that there is some reversibility of the neurological problems after liver transplantation. In children, the main cause is biliary atresia. Again, the liver transplantation is the only treatment, but it's sure now that there is an irreversibility of the neurological problems, meaning that kids who have a chronic HE also show learning disabilities, cognitive deficits, and mental retardation after liver transplantation, something that was showed also by Valerie McLean in the UK. Then we are wondering if there is something that makes children more vulnerable. So what are the motivations of the studies and what are the unknowns? So in the adult brain, the precise metabolic changes and the mechanism which are leading to the disease are not known. We don't know what are the chronological events, we don't know what are their precise timings, and we don't know the early changes happening in this disease. And this is because we only have a few studies and they're all at the later time points. In kids, we know that there are some neurocognitive problems that seems to persist after liver transplantation. Olivier Bresson showed, using the 3D cell cultures that I previously showed you, that ammonium exposure impairs axonal growth in the developing cells, brain cells. And we believe that pediatric brain might be more vulnerable to the complications of chronic liver disease. But the current therapies are aiming at lowering peripheral ammonium, and they are not sufficient. So they are not avoiding the long-term clinical consequences in the children. Therefore, identifying novel molecular mechanisms and novel treatments would be a major advance. So now let's just go to the results that we obtain in the developing brain and comparing them to the adult brain. So we use an animal model of bile duct ligation in rats. It's a model which is approved by the International Society of Hepatic Encephalopathy and Nitrogen Metabolism. It's the only model accepted for now. The bile duct ligation was performed by isolating the bile duct, the common bile duct. It was triple ligated and sectioned, as shown here in the image. We performed the surgical ligation, then the chronic liver disease is developing, the animals become yellow, they have hyperammonemia, and then we have also development of the neurological disease, what we call hepatic encephalopathy. So our aim was to use the advantages of high-field proton and phosphorus magnetic resonance spectroscopy to analyze the longitudinal changes in the large number of metabolites. During the progression of chronic hepatic encephalopathy, we can follow up to 17 brain metabolites with localized proton spectroscopy at short echo times and the high magnetic field, the 9.4 Tesla. We focus on the energy and phospholipid metabolites using phosphorus. We also focus on early changes by performing our longitudinal studies, and we hope to get some insights into the chronology of events leading to hepatic encephalopathy. We are also wondering why the young chronic liver disease rats suffer from more severe and different neurometabolic changes. And we are using a multimodal approach. In addition to the proton spectroscopy and phosphorus spectroscopy and imaging studies, we perform behavioral tests, blood testing, histological measurements, as done by Kasia in the lab, FDG PET experiments done by Bernard and Jesse, and EPR experiments for the measurement of oxidative stress, again done by Kasia. So today, there are no similar studies performed in animal models or children with chronic hepatic encephalopathy. So the study design, we have adult rats which have the surgery at the week 0. Then they are followed longitudinally every 2 weeks, and also scan is performed before the surgery, and all the data are compared with the scan 0. So it's an adult brain, so we are not expecting to see differences over the time. So the main differences that we will see, it's because of the disease. And then we use rats which have a surgery at the postnatal day 21, which is equivalent with a 9-month-old human brain. So the rats have the surgery at week 0 because they are still in development. They don't have a scan at week 0. They only have scans at week 2, 4, 6, and 8, and they're always compared with sham animals at the corresponding age. So now just to see how the experiments are done. So we are using the 9.4 Tesla. We are using a home-built coil, and here it's a voxel where we acquire the data in the hippocampus. We are using the special sequence with an echo time of 2.8 milliseconds, LC model for quantification, and water as internal reference. And here you can see an example of a spectrum acquired at 9.4 Tesla. For the phosphorus spectroscopy, it was developed by Veronica, a PhD student working in our group during her PhD. So here you can see an example of what we get. You see the voxel, the coil, which was built by her. The localization was a little bit changed compared with an additional traditional sequence, meaning that we are doing outer volume suppression in X and Z direction, and we are using an ISIS module in the Y direction. We are exciting with an adiabatic half passage pulse, and we added NOE enhancement and decoupling to enhance the signal obtained in the phosphorus. And of course, we don't have water here to use it as an internal reference. So we use a phosphocreatine from a voxel and from an acquisition performed before using proton spectroscopy in a similar voxel. So this is a quality of a spectrum that we acquired at 9.4 Tesla in something like 50 minutes. You can see the phosphomonoesters, the phosphodiesters, the inorganic phosphate, the intracellular, and what we believe that it's extracellular, and the oxidative, the metabolites involved in the oxidative stress, in addition, of course, to the phosphocreatine and the ATP resonances. So we first looked into what's happening in the adult brain. So we took adult rats, we performed the surgery, we measured them in the hippocampus longitudinally. Here is an example of spectra that we're obtaining at scan 0 and scan 8, but we scanned them at 0, 2, 4, 6, and 8 weeks, as shown previously. So you can already see in the spectra that there is a pronounced increase in the brain glutamine and a decrease in the osmolytes, inositol. So we obtained for the first time, we saw longitudinal changes and very early changes like an increase of the glutamine in the rats at 2 weeks already after surgery. We saw a decrease of creatine as an osmotic stress to the glutamine increase, again very early at 2 weeks after the surgery, and a decrease of the antioxidant ascorbate, which is a response to the ammonium load. And then, of course, these changes continued over the disease evolution, and we had the additional changes in the osmolytes, like inositol, taurine, total choline, which were evidence of an osmotic stress combined with an oxidative stress. Kasia also performed experiments. She stained the brains with GFAP, and she showed that this osmotic stress and ammonium probably leads to a change in the astrocytes morphology, like a decrease in the processes, the number of the processes, and the length of the processes. So now if we look to what happens in the P21 rats, so in the rats who had surgery at P21, which is equivalent to 9 months old humans, and to rats who had the surgery at being adult rats. We first look into the plasma ammonium. If you remember, it's increasing because of the bile duct ligation, and we look at the plasma bilirubin, which is a sign. Its increase is a sign that the surgery was well performed. So if you can see that we have an increase of plasma ammonium, which is much more stronger in the P21 rats, in the rats operated at P21, which longitudinally become older, of course, and also an increase in plasma bilirubin in the developing brain. So this makes us believe that the liver disease is stronger. So the PAPs are more, or they have a stronger disease. So they are, how can I say it? They have a stronger chronic liver disease. So their liver is strongly affected than the adults. But this is not the main cause of the changes that we see. So if we look in the spectra, these are the spectra in an adult rat at before the surgery at week 0 and at 8 weeks after surgery. You can see again the changes in glutamine, the increase in glutamine, the decrease in inositol as an osmotic response. And in the young rats, you can see from the spectrum already that glutamine is much more higher here than here. And you can also see that inositol is strongly decreased compared with the adult rats. So now if you look at the brain glutamine, in orange you have the adult rats, and in blue you have the pups. You can see that the increase in the adult rats, it's around 139%, while in the pups it's 370% compared with their corresponding shams. And if we look at the correlation between the brain glutamine changes and the plasma ammonium changes, we can see that the slope is steeper in the pups than in the adults. So this makes us believe that it's not only the fact that the pups are sicker than the adults, that's the main cause. We also believe that the effect, that there's a stronger effect of the ammonium during the brain maturation, and this can be due to the changes due to the glutamine synthetase maturation, which is not the same. So in adults it's maturated, this enzyme, while in pups it's not yet reaching this. So the osmolytes, we also see that they react differently, meaning that in the adult brain we have in the pups we have a stronger decrease on the osmolytes in blue here compared with purple, with orange, which is of course expected because we have a stronger brain glutamine increase. So the osmotic answer in the young cirrhotic rats is faster and stronger. You can see that the decrease is even faster than in the adult brains. So then if we look at the other metabolites, all the other metabolites like energy metabolites, total creatine, creatine, phosphocreatine, and lactate, they show a stronger pattern than in the developing brain than in the adult brain, meaning that the changes occur faster and they are stronger than in the adult brain. Then also the neurotransmitters, it's the same thing. Glutamate and aspartate, the decrease is stronger in the developing brain than in the adult brain, and also for the antioxidant ascorbate, same story. So adult and young cirrhotic rats, they show changes in plasma parameters, they show an increase in brain glutamine, they show a decrease in the other organic osmolytes as a response to the increase of brain glutamine. And they also show neurotransmitter perturbations and antioxidant perturbations, so changes in these metabolites. But the young BDL rats, they showed an increased vulnerability to metabolic insults of chronic liver disease. They showed a similar pattern to the adult rats, but their changes were stronger in the osmolytes, neurotransmitter, antioxidants. They appeared earlier, like glutamine, minoxidil, taurine, glutamate. And there are some changes which are unique for the developing brain, like the changes in aspartate, lactate, and phosphocreatine, which do not appear in the adult brain. And also the energy metabolism is more affected in the brain, in the developing brain, than in the adult brain. So now just some preliminary results in children with chronic liver disease, a study that we've performed at 7 test labs. So what were the motivations of the study? The fact that there are very few MRI studies performed in children with chronic liver disease, and they are limited by the low magnetic field. 1.5 Tesla, which does not allow the separation between glutamine and glutamate. And as you've seen previously, they're very important metabolites in this disease. We saw that the young VDL rats show stronger changes during the brain development. So we hypothesize that chronic liver disease kids will show additional and stronger brain metabolic changes also. We use the 7 Tesla. We use the advantages of the 7 Tesla that we have at the CIBM to allow us to identify changes in glutamine and glutamate separately. Something that what I said that is not possible to see in at low magnetic fields. And we also wanted to see if we are able to identify additional low concentrated and overlapping metabolites like GABA, aspartate, ascorbate, and so on. So as I said, we use the advantages of the increased resolution provided by the 7 Tesla to get the full metabolic profile, to do brain volumetry and T1 relaxation times in children with chronic liver disease and congenital portosystemic shunts, which are a type B HE. So we had children between 8 and 16 years old, which showed chronic liver disease or congenital portosystemic shunts. They had a proton MRI and MRS at 7 Tesla in the CIBM at EPFL. We performed a 3D MP2RAGE sequence, which was used for an automatic segmentation of the deep brain structures using the Morphobox from Siemens. So all the data, all the imaging data were processed by the Siemens group. And we obtained absolute volumes and averaged T1 values were calculated for the basal ganglia and each deep nucleus individually, like caudate, putamen, thalamus, pallidum, and then we measured in the hippocampus and amygdala. For the proton spectroscopy, we used a semi-adiabatic special sequence at an echo time of 16 milliseconds, and we had 2 voxels, one in prefrontal cortex dominated gray matter, and in the other one in the same brain region, but with more white matter. The metabolites were quantified using Gaussian model and water as an internal reference, and we also had some neurocognitive testings and routine labs which were performed in these children. So we had 6 patients with chronic liver disease. We had 5 of them with chronic liver disease, 2 were girls, and 1 was with a congenital portosystemic shunt. The causes of chronic liver disease are quite diverse. We have congenital disorders of glycosylation. We have progressive familiar intrahepatic cholestasis type B, autoimmune hepatitis. We have some results, some plasma and serum results, meaning that the mean serum bilirubin was normal in this animal, in these patients, and the mean plasma ammonium was 26 micromole per liter. We had also 20 controls, and we had 10 girls and 10 boys. So these are the main results. So this is a screenshot from the volumetry, which was performed by Benedict. You can see here the MP2RAGE unit sequence, then the T1 maps, and an example of brain volumetry. So here are the absolute volumes for the controls in black, for the 5 chronic liver disease kids in darker green, and for the 1 girl with a congenital portosystemic shunt. As you can see, between the controls and the chronic liver disease, these 2 colors, there is no difference. However, the congenital portosystemic shunt girl shows a decrease in the majority of the brain volumes. Then if we go at the mean T1 values we were expecting, so in chronic HE, in type C HE, due to the bilirubin accumulation, we are expecting to see a change in the T1 values in the basal ganglia. We were not able to see, due to the manganese depositions, which are due to the bilirubin accumulation, we are not able to see these changes in the T1 values, which is maybe also normal because all our children with chronic liver disease, they had a compensated chronic liver disease and they showed no change in the bilirubin values. So this is an example of a spectrum that we acquired in the gray matter in these children. And you can see down the control spectrum, the chronic liver disease spectrum, and the congenital portosystemic shunt spectrum. So visually, the chronic liver disease and the controls, they're very similar, while in the congenital portosystemic shunt, we see an increase of the glutamine and a decrease of the main osmolytes. So now if we go to the results, like for the brain volumetry, the control and the chronic liver disease in the gray matter show no differences whatsoever in any of the brain metabolites. However, the congenital portosystemic shunt girl shows a high increase in the brain glutamine. It shows a decrease of the brain osmolytes like inositol, taurine, total choline, and even total creatine, something expected due to the increase of the brain glutamine. What is quite interesting is that we saw changes in the GABA, decrease of glutamate, and even a decrease of N-acetyl aspartate and glutathione. So these are all novel findings. So to conclude, in patients with compensated chronic liver disease, we see no significant metabolic alterations. In congenital portosystemic shunts, the neurometabolic changes were more pronounced, meaning that we have a decrease in the, in addition to the decreases in the measured brain volumes, which are likely related to the impaired neurocognitive functioning. And in a type B hepatic encephalopathy, the congenital portosystemic shunt, the brain is likely exposed to a higher load of neurotoxic substances than in patients who have some degree of porter flow, like in type CHE. Just to remind you that our chronic liver disease patients, they have a compensated chronic liver disease, meaning that they have more or less a normal life. So in young BDL rats, we saw an increased vulnerability to the metabolic insults compared to adults. We saw a similar pattern of neurometabolic changes like in the adult rats with chronic liver disease, but the changes were stronger. The changes appeared earlier, like in glutamine, osmolytes, and neurotransmitter glutamate, and some changes were unique for the developing brain, like changes in aspartate, lactate, phosphocreatine, and glutathione. So all these are neurotransmitters, metabolites involved in energy metabolism and in the oxidative stress. We had a seriously altered phospholipid and energy metabolism, like measured with phosphorus MRS, a decrease of creatine kinase rate, and decrease of phosphocreatine and ATP, and an increase of lactate. We also performed treatments in bile duct-ligated rats, and they are in the next slide. And we showed that high creatine diet leads to a partial recovery of brain creatine. So brain creatine was decreased, total creatine was decreased in young BDL rats. Providing them a high creatine diet restores these creatine levels, and we obtain a positive, we saw a positive, a protective effect on some neurometabolites like glutamine, ATP, ascorbate, and glycerophosphatidylonamine. In the pediatric patients, the type B HE, the brain is likely more exposed to a higher load of neurotoxic substances. We see stronger neurometabolic changes and a marked decrease in all measured brain volumes. Just to finalize with some ongoing and future studies. So as I said, we are treating our rats now with creatine, with probiotics, antibiotics. Probiotics, we are using vivo mix in a collaboration with Professor De Simone in Italy. So why we do it? Because we saw that there are neurocognitive problems which persist in kids after liver transplantation. We showed stronger changes in total creatine in BDL rats, and especially in the young ones. And Olivier Bresson showed in vitro that ammonium downregulates creatine synthesis through a decrease through the agat enzyme. And creatine diet, it's used as a neuroprotective treatment in many diseases. So these are some spectra acquired in our pups during the creatine treatment. So you have the sham with creatine, the BDL rats, and the BDL rats with creatine. These are proton spectra. And here you have the corresponding phosphorus spectra. And as I showed you previously, we have some neurometabolic changes which are reversed by the high creatine diet in the young rats, meaning that the energy metabolites, total creatine, and ATP are restored. So we have a decrease in the BDL rats, and while we provide the treatment, this decrease is restored. We have the antioxidant ascorbate, which also recovers more or less its normal values. If you see here, you can see the glutamine in the sham animals. You can see it's increased visual in the BDL rats. And if you look at the BDL rats treated with creatine, you can see that the increase of the glutamine is less pronounced. And then we have also changes or restoration of the phospholipid metabolism. So these results are promising, and we are testing other different molecules. And in the same time, Kasia and Dunya, they are working on the brain regional difference. So we would like to see if there is a certain brain regional difference in chronic hepatic encephalopathy. It is not known. So what we did is that we took different brain regions like the striatum, hippocampus, and cerebellum. We performed proton spectroscopy, and we saw that the increase of the glutamine is stronger in the cerebellum compared with the hippocampus and compared with the striatum. And we also saw differences. Here it's a Golgi-Cox staining. We saw differences in neurons between hippocampus and the cerebellum. You can see here the measurements. So all these are ongoing studies. So now I'm finishing up. So I would like to thank you for your attention. And I would like to thank the people who are working very closely on this project. They're all listed here. Oops. They're all listed here. We have Veronica who left. We have Dario who's doing the surgery of all our rats. We have Stefan who's helping. We have Dunya, Kasia who performed experiments, Emma who performed experiments, Bernard who did the PET experiments. You have there Jessie who newly joined our team, and Olivier and Valerie. And thank you all for all your attention.
Okay, thank you.
Thank you, Cristina. I was on mute. I was saying, I think we can give her a hand of applause. I don't know if we can do this, but.
Oh, thank you. And yeah, before closing, I would like to thank some collaborators who are not present today, who are on in the hospitals now, working strongly, hardly. So thank you for their hard work. And thank you all for being here on all these very special times.
Okay, so I think we're way beyond the time, but I think we're okay with at least one question from the audience.
May I ask you a question?
Yes.
Thank you first. It was very nice presentation. So there was a lot of information, but in the past we worked on this hepatic encephalopathy, and there was this hypothesis of manganese together with motor symptoms.
Yeah, the Parkinsonians.
The T1 in the pallidum. Is it, I mean, with respect to this older, let's say, older hypothesis, what is the actual status?
So we can see the changes. So indeed, there's a manganese deposition in chronic liver disease children. Valerie performed with Sylvian. They performed a study on kids at 1.5 Tesla in Geneva in the HUG. And they saw the presence of these changes in the T1, so decrease of the T1 in the basal ganglia. We were not able to see it in our children, maybe because they are not so sick. In the adult rats or in the pups, we've never performed an MP2 rate sequence. It's something that I would like to do with Ileana. And yeah, and then Kasia is also trying, she took some brain samples and she's trying to measure with EPR, electron paramagnetic resonance, on these samples, the manganese deposition, the copper deposition, and so on. So it's something that we are investigating, and we know that it exists, but when we did the the experiment, sorry, on the kids, we haven't seen it, and on the rats, we haven't done it yet.
Mm-hmm. And with respect to the choline changes,
yeah,
what is your hypothesis? Do you see similar changes in the phosphorus spectrum, like phosphocholine or glycerol phosphocholine?
yeah, we see the changes. So so they are confirmed. So we believe that these changes are due to the increase of the brain glutamine. So glutamine is increasing in astrocytes, mainly in the astrocytes because of the glutamine synthetase enzyme, which is located in the astrocytes and who's converting ammonium into the glutamine. So we believe that this glutamine increase will lead to an osmotic stress, and all the other osmolytes will leave the cell to prevent the swelling. If it's prevented or not, it's not yet sure. And we saw these changes in the decrease of the total choline starting 6 to 8 weeks in the adult rats, and even earlier 4 weeks in the pups. And we confirm it with the phosphorus data. And we believe that in the young rats, what Veronica did and what she just submitted, it's in the under review her paper, it's that these changes in the total choline are very important in the developing brain, because total choline is very important for the, if I remember correctly, for the myelination. And and we have 70% of decrease, or around 70% of decrease of total choline during brain development. So this might explain or work together with the neurocognitive deficits that we see in the patients, in the young patients.
Okay, thank you.
Okay.
You're more than welcome.
Thank you, François.